And the idea was, during the Zika virus or epidemics, there was groups that were able to use human brain organoids to model Zika virus effect on neural tissues. We wanted to see if SARS-CoV-2 can be affecting neural tissues. So naturally, once we got a good infection going, we asked the most basic question: can SARS-CoV-2 infection result in cell death? And overwhelmingly, we immediately saw this, huge infection along with a lot of cell death marked here in green, right, by the tunnel staining. And we were extremely excited. But what we were really kind of, shocked to see was that the cells that were infected, shown in red here, were not the cells that were dying. In fact, you can see here, there's almost virtually no overlap between the cells that are marked in red, right, the infected cells, and the cells that are going through cell death, the green tunnel positive cells. When we quantify this, we were, overwhelmingly in the whole organoid, we were able to see that most of the tunnel positive cells were not infected with SARS-CoV-2, giving us this idea that dying cells were not infected with SARS-CoV-2. And even within a single organoid, in different regions of the organoid, you can see here very clearly that SARS-CoV-2 infected cells are neighbored by a lot of tunnel positive cells, shown in the yellow box, compared to the white box, where if there is no infection happening, cell death is also not occurring in those regions. So to better kind of probe this question of what's actually going on and why are the cells that are not infected actually dying in these organoids, we decided to use single-cell RNA sequencing to answer this question. So by using single-cell sequencing of the human brain organoid, we were able to come up with a variety of different cell types in the organoid. In addition, we were able to clearly detect which cells were infected by looking for the SARS-CoV-2 mRNA. And you can see here, 96 hours after infection, we had a large amount of organoids or cells in the organoids infected with SARS-CoV-2. So to see what was happening transcriptionally after we infected them, we first decided to compare it with a very canonical neurotropic virus, like we talked about, the Zika virus. And to our surprise, what we noticed was that the genes that are upregulated in Zika virus the genes that are upregulated in the organoid after Zika virus infection virtually had no overlap with the genes that were upregulated after a SARS-CoV-2 infection, shown here in the bottom. In fact, if we take the genes and look at the pathways that are involved in these two infection models, they were very, very different. We have on the top kind of shared gene signature of some sort of, foreign infection occurring, right? Foreign stress or foreign stimuli. However, while Zika virus was able to upregulate pathways related to kind of type 1 interferons, innate immune responses, these canonical immune responses that we expect to see, the SARS-CoV-2 infected neuronal cells actually showed none of these pathways. And rather, they were going through cell cycle pathways and more metabolically active pathways. To even look at it more granularly, we took the cells that were actually infected in a specific organoid and compared it to the neighboring cells, right? The cells that were not infected, but in the infected conditions. In the cells that were infected by SARS-CoV-2, we were able to see that they were going through a hyperoxic pathway, along with increase of many different metabolic pathways. And the neighboring cells were actually going through a response of hypoxia, and they were having decreased oxygen levels and going through cell death. And the cell death we noticed, right, from the previous things. And to make sure that this was indeed truly happening, we validated our findings by staining for different markers of hypoxia. And here you can see that when SARS-CoV-2 infection occurred, the local environment in the organoid became much more hypoxic, as you can see here by the hydroxy probe that we used. So we now had a kind of a working model of what might be going on. The SARS-CoV-2 infected these neuronal cells that actually didn't result in death, but increased in metabolic activity to replicate itself. And by doing that, the cells then were sequestering the nutrients needed to survive from neighboring cells, and the neighboring cells were the ones that were actually dying from the consequence of the infection. This is all very exciting and interesting, but as you know, organoids don't have the capability to have all the cells that we would normally find in a normal central nervous system. So we decided to adopt a couple of different models of SARS-CoV-2 for mice. And very quickly, we were able to actually see that SARS-CoV-2 had a huge neuroinvasive potential even in mice. Of course, the mice require exogenous ACE2, human ACE2 expression. But once the receptor expression is present, the cells are able to harbor these viruses and allow them to replicate at large amounts. And you can see many neurons in different parts of the brain could be infected, as seen here. And so this is an iDisco imaging that one of our collaborators, Nico from Paris, helped us with. And you can see the cerebellum is mostly spared in this mice, but all of the other parts of the brain are highly infected. And using Nico's iDisco method, he was also able to map out the vasculature that was present in the brains of the mice. And we thought that the vasculature, if there was truly this hypoxic kind of response that was occurring from SARS-CoV-2 infection, then the vasculature might actually be also changing in the brains of these mice. We were able to calculate the densities of the mice, the vessels in the mice, and what we saw was after infection, the vessel densities were changed significantly. And this also was very much aligned with hotspots of virus infection. Very similar to what we saw in the organoid. So we were very excited about this idea, and we have kind of ongoing projects on kind of looking at this further. And then finally, at the same time, there was this big need and a big excitement to see if the virus can actually infect human brain tissue. So it was hard for us to get tissues, but our collaborators, once again from France, really pulled through. And they were able to obtain patient samples who passed away from SARS-CoV-2. And what we saw was that in certain patients' brains, we were able to see SARS-CoV-2 positive neurons. And what was most interesting to us about this was that in other brain infections, such as like herpes virus or Zika virus or West Nile virus, we tend to see this huge influx of lymphocytes or other immune cells that are contributing to the damage, right, while it's clearing the brain. However, in the SARS-CoV-2 infected brains, we didn't see the same sort of huge inflammatory response. We saw infected neurons, but a different type of kind of follow-up and response that occurred. And this is consistent with like what other groups have seen. We saw these regions of microinfarcts and hemorrhages that were resembling kind of possible mini strokes. And what we saw was that in these regions of microinfarcts, we also saw higher rate or higher amounts of spike SARS-CoV-2 staining. And it was very reminiscent of kind of what we were seeing in both the mouse model and the organoid model. And although we haven't been able to completely follow up with a mechanistic understanding of why or how this is happening in patients, I know a lot of groups have different interesting hypotheses and they're working on it. So I think it's a very exciting time for this kind of research.
All right, Karen Thacker, Columbia Medical Center. Thank you so much. It's been a pleasure talking to you.
Yeah, thanks for having me.
Thank you.
It's been a great time. Yeah,
Terrific.
Thanks everybody.
Okay, bye-bye.
Bye.
So, bottom line, they don't find virus in the brain.
No.
Yeah, all the stuff they see is probably due to hypoxia and inflammatory cytokines. And
Right.
Right, which